You might be wondering, why didn't they just stick to common names? Why bother with all the complicated Latin? Well, a formal classification system is absolutely crucial. It allows us to study collective groups efficiently, it shows evolutionary relationships over time, and it lets us correctly identify species without pesky geographical language barriers getting in the way.
Linnaeus came up with a brilliant solution to this naming headache: binomial nomenclature. It's essentially a two-part naming system that instantly tells us which organisms are closely related enough to share a genus, but still distinct enough to be separate species. The rule here is strict but elegant. The genus is always capitalized, the species is lowercase, and together they give us super precise scientific names. Think Homo sapiens for us humans, or Zea mays for the maize plant.
To organize all these names, we use this sort of inverted pyramid of taxonomic hierarchy. It starts really broad up at the kingdom level and gets incredibly specific all the way down at the species level. And to remember this exact order, you just need this fantastic mnemonic. Keep pot clean, otherwise family gets sick. Kingdom, phylum, class, order, family, genus, species. Easy, right?
Moving on to Section 2, the 5 Great Kingdoms based on Robert Whittaker's 1969 system.
Back in 1969, Robert Whittaker brilliantly sorted life into 5 main drawers. He looked at body complexity, how organisms get their food, and most importantly, the core structure of their cells. So on one side, we have prokaryotes. These are simple cells that lack a distinct nuclear envelope in those membrane-bound organelles. On the flip side are eukaryotes. These are much more complex, boasting a true, distinct nucleus and complex double-membrane organelles like mitochondria.
By using those exact cell rules and factoring in how they eat, life gets perfectly sorted out. We've got Monera, the simple unicellular prokaryotes like bacteria. Then Protista, which are unicellular eukaryotes. Next up is fungi, which are multicellular, have cell walls, but they're saprophytic, meaning they eat by absorbing dead matter. Then we have Plantae, the autotrophs that make their own food. And finally, Animalia, heterotrophs that don't have cell walls and actually have to consume other organisms to survive.
That brings us to Section 3, the plant family tree, where we'll be decoding Kingdom Plantae.
The plant kingdom is this massive group of autotrophic, multicellular eukaryotes tightly wrapped in rigid cellulose walls. But what's really fascinating here is how they evolved in complexity. We literally start in the water with simple aquatic algae that lack true roots or stems. Then we step onto land with bryophytes. These are often called the amphibians of plants because they still desperately need water to reproduce. Finally, we reach the tracheophytes. These guys totally conquered the land by developing specialized vascular tissue, xylem to transport water and phloem to transport food.
And within those highly successful tracheophytes, we see 3 major subdivisions. First, you've got the fern-like pteridophytes that reproduce using spores hidden in structures called sori. Next are the gymnosperms. These are your pines and conifers that protect naked seeds inside tough cones. And lastly, the most advanced group of all, the angiosperms, boasting true flowers and keeping their seeds safely covered inside fruits.
To give you a sense of just how incredibly successful that covered seed and flowering strategy really is, angiosperms encompass about 300,000 distinct species. That makes up a staggering 80% of the entire plant kingdom. They absolutely dominate the modern planet.
Now, those dominant angiosperms are further split into 2 distinct groups based on their seed leaves, known as cotyledons. Monocots, like a banana or bamboo, have 1 seed leaf, parallel leaf veins, fibrous roots, and often hollow stems. Dicots, on the other hand, like a mango or a rose, have 2 seed leaves, net-like reticulate veins, a strong central taproot, and solid woody stems. Seriously, once you know this, you're going to look at every leaf and root outside totally differently.
All right, let's jump into our final section, Section 4, Blueprint of the Animals, Unlocking Kingdom Animalia. Kingdom Animalia is a massive collection of eukaryotic, multicellular heterotrophs that notably lack cell walls, which gives them incredible flexibility. To remember the evolutionary order of the 9 major animal phyla, scaling from the simplest invertebrates right up to the vertebrates, rely on this fantastic mnemonic. Please catch pretty apples and also make excellent cake. Porifera, Coolentera, Platyhelminths, Ashhelminths, Annelida, Arthropoda, Mollusca, Echinodermata, and Chordata. But before we categorize all these animals, we really need to understand their structural building blocks. Life in the animal kingdom scales up elegantly. It ranges from a simple cellular grade, where cells just kind of work independently, up to a tissue grade,
then to distinct organs, and finally to highly complex organ systems where multiple organs work together to keep the animal alive.
We also group animals by their physical symmetry. So we have asymmetrical animals with no mirror planes, radially symmetrical animals like starfish, and bilaterally symmetrical animals like deer or humans. Interestingly enough, the octopus actually has bilateral symmetry overall if you look at its entire body, even though its tentacles alone exhibit a radial pattern. Nature is just wild like that.
Embryos actually tell us a whole lot about an animal's blueprint too. As embryos develop, they form germinal layers that eventually become the organs. Diploblastic animals only have two layers, an outer ectoderm and an inner endoderm. More complex, triploblastic animals add a crucial third middle layer, called the mesoderm.
That mesoderm layer is incredibly important because it dictates whether an animal has a true coelom. A true coelom is a body cavity completely lined by that mesoderm. If an animal has this, it's a true coelomate. If the cavity isn't entirely lined by mesoderm, it's a false pseudocoelom. And if there's no cavity at all, there's simply a coelomate. Let's see how all this terminology builds our very first, simplest phylum, Porifera, better known as the sponges. Here we're looking at simple, asymmetrical, diploblastic animals with just a cellular grade of organization. They literally don't have tissues. Instead, they rely entirely on specialized collar cells or choanocytes. These cells have tiny flagella that constantly pump water in through small pores called ostia and out through a large pore called an osculum, catching food and oxygen along the way. From the simplest sponge pumping water to the massive diversity of angiosperms dominating the land, this filing system brings beautiful, logical order to the natural world. But as we uncover more about the bizarre and wonderful organisms sharing our planet, we have to ask ourselves, is this map of life complete? Or with every single new discovery, are we really just getting started? Definitely something to think about next time you step outside.